again, we're keeping it kind of simplistic by just assigning letters and not worrying about allele types or the 3-letter code, so just keeping it very simple. And you should right off the bat see that some of the letters are repeated, okay? So the reason is, so you've been given these 4 strains, but they were all derived from the same original strain. Okay, so all of these HFR strains came from the original F+ strain. So this is all information you would be given to do this problem. And ultimately, it's asking what the order is of these genes or markers. Okay, so this comes down to understanding too the biology behind how you make an HFR cell. Okay? So we originally make an HFR from an F+ whose F plasmid integrated in with its chromosome, right? So that's what it means by they all came from the original same F+ strain. So you're trying to basically recreate a genetic map for this original F+ strain. That's what you're asking. Where do these markers fall? What order are they in as we go around this circle? So the way to approach a problem like this is how I would do it. Some folks, again, you don't have to break it down this way. You'll find different ways that work for you. But I would look at each individual strain and make my own little map for that. So you'll end up with 4 maps here, and then you can consolidate them, because they're going to overlap with each other. Some people might be able to see that already as they just look at the letters up on the board. Okay, so you can kind of start with strain 1. And you know there's a Q, W, D, M, and a T. Okay, that's all information you have just from that strain 1. So you're essentially just taking the order of these markers, because they're shown in the order donated, and you're placing them around a circular structure. Okay, you're picturing it all being on one chromosome. Do the same thing for 2. Okay. 2 starts at A, but you have an overlap here of T and M, right? So if we kind of keep, just so that you can see the picture better, if we keep T and M in the same, somewhat the same location, if this was a clock, right, you're going around, kind of keep them at the same time or whatever, and then you can add your other markers on there. And so in this case, I'm actually walking backwards from M, T, P, X, and A. So if they're entering in the order shown, that's the order that they were actually donated in, this one is entering with the Q coming in first, right? So often with these maps, you can indicate the order that this strain is rolling in, okay? That these markers, these genes are kind of coming in as. So with strain 2, where would I put my little arrow? In front of the A, right? Because A is the first one coming in. So to help you kind of understand which direction you're flowing in, you put it right in front of that first marker, just like it was the front of a train or the front of a snake or whatever it is you're thinking about as it's moving its way into the next cell. That's the front. That's the first thing to come in. for 3, what are the two letters that overlap on 3?
A and S.
Yeah. So X, A, and I'll kind of keep them still in the same spot just so it's easier to consolidate them. And then you have C
And B.
and, thank you. And B in this case is the first one to enter. So you add your arrow heading out in that direction. Is this making sense how I'm walking through this? Same thing for 4. Starts with
B.
E, and then Q is on there,
W.
and then around, so yeah, W, B, and N. And in this case, B is the so that's how I do these problems. If you can look at this set of letters and you just instantly see the map, then go for that. But I usually break it down and draw each map and then merge them. Okay? You're basically consolidating those 4 maps without, because there's lots of overlap between the letters. So no letters is represented on there twice. You just can add in all of your alleles, like, or markers, I'm sorry, Q, W, B, M, T, E, X, and A, B, and C. I have a lot more that I missed.
And B. C and
And your answer can be if you picked this circle up and then rotated it around, okay, that's still correct. Or if you kind of turn this clock, that would be correct on an exam. So long as you have the order correct and the sequence, that's what we're looking for. Okay, so if you're drawing, if you approach this problem and your drawing's kind of shifted, that's completely fine. Because this is, right, a 3-dimensional circular piece of DNA that's moving all around and can flip over and all that kind of stuff. Okay. So those are still the correct answer, but you're trying to get this order as it goes around. Okay, so that's how I would approach this type of problem. Make separate maps for each strain and then consolidate them all into one map. And look at, that's it, blank page. So, is there any questions about the type problem we just walked through? So that's what I have for you guys tonight. Wednesday, you will get your exams back. We'll spend some time talking about it, and we'll keep talking about our bacterial genetics and stuff.
you can keep that.
This time?